You mean restaurant? No. I have closed makeup.
Okay, guys, let's get started. Who ate too much pumpkin pie? I did. I feel it. Oh my god. I got to make 2 pumpkin pies. I know you guys are excited. One was dairy free, not as good. Just saying. Okay. So today we're going to start our unit on cell cycle. Before we do that, 2 notes. Exams by Friday. I'm sorry. I had a very busy. I had 4 kids I had to take care of on, what day was that? On Thursday or Wednesday? I don't know what day it was. 4 kids, all under the age of 5. Yeah, not all of them were mine, God's sake. So anyways, no excuses. The average on the multiple choice was fantastic, and it has been correlating. It was like up in the 80s. So, and the ones that I have graded so far have all been up in the 80s, 90s, and over 100. So I'm hoping we're going to replicate what we did on that last exam. So that means either you guys have somehow gotten a hold of my old exams, or, and in that case, I just don't care, or you guys are actually figuring out how to study for my exams and doing better, which is great. Other thing, quiz 11, there was an error. A number of people emailed me about checking. There was one that was multiple answer instead of multiple choice. I hit the wrong question type. And so if you hit everything including all, you got it wrong. If you hit all alone, you got it right. Thank you for letting me know. I've graded it manually, so all of you who truly got it right. Please always. Some people are very apologetic when they email me about that, but if something's wrong, something's wrong. So, that's it for now. Okay, cell cycle. So that's just basically the copying of the genome and organelles and passing them on to daughter cells. That's a cell cycle, right? You can call it a lot of fancy things like mitosis and DNA replication. But really, you have to copy your genome, you have to replicate everything and separate it in 2, and that's how the organism survives, right? So this is not the most traditional picture. You've seen the next one is what you normally see, but you can think of it as chromosome replication happens here, then you get chromosome segregation, cell division, 2 daughter cells, right? So this leads to this question from this weekend's quiz, right? The following events are key activities of the cell cycle. Which one is wrong? None of them, right? So a lot of people miss the organelle duplication because you don't think of it as an obvious thing, but you do need to, right? Right? Because the daughter cell needs organelles too, not just DNA, right? So cytokinesis, right? The splitting of the cells, DNA replication, chromosome segregation, not in that order. If you do cytokinesis before DNA replication, that's wrong, right? Yeah, it's a problem. Okay. The major elements of the cell cycle are chromosome replication and distribution to daughter cells. So that would be S phase for DNA synthesis and M phase. Right? So you get your replication here and then separation. Very simple. We start simple. Don't worry, it'll get harder. I promise. So DNA is replicated towards the end of interphase. Again, there's been a lot of confusion throughout the history of cell biology and students learning cell biology about what interphase is. It's basically the whole cell cycle that is not mitosis. And if you look at this, right, here's your M phase, and the rest is interphase. And towards the end of interphase, you do have DNA replication. So DNA replication occurs towards the end of M phase. If all goes well, what do I mean by if all goes well? What could go wrong? What could go wrong that might prevent progression to M phase mitosis? Yes. Right, so incomplete duplication of chromosomes, right? You don't want that to happen. You don't want to separate your chromosomes if they're not completely duplicated, right? Makes sense. What else could go wrong? Yeah. So not the right environmental conditions. So that's like a vague thing, which is true. It's in the textbook and everything. What does that mean? Not in poor environment. I know. Yeah. So if your mitotic spindle hasn't set up, right? Right. So that's one thing. Yep. Or it could just be the environment, like the growth factors necessary are not present, right? What else? What else could go wrong that would prevent progression to M phase? Yes. Lack of nutrients. Yeah. What else?
A breakdown in signaling.
Some kind of breakdown in signaling. So what are you thinking of?
Any necessary transcription factors for one thing.
So like if some kind of factor that's required for activation of mitosis, of progression to mitosis isn't there? Okay, yep, why not? That makes sense. Yep. Anything else? Yes. Right, right, well that actually, DNA condensation is in the beginning. That's happening right over there. Prophase, prometaphase. So you're already in, but that will absolutely prevent metaphase anaphase transition. So you're right that it won't complete. What else? What else? I'm thinking of one more thing that's going to prevent progression to M phase. DNA damage. If your DNA is damaged in some way, right? That will result in a stall actually in DNA replication because you'll have stalled replication forks. Okay, right. So if all goes well, all those things don't happen, you go to M phase and you'll get your chromosomes segregated to daughter cells as a result of mitosis. So here's your classical picture that everyone sees. You say cell cycle, this is what you see, right? This is your M phases with your gap phases, right? Mitosis, G1, S phase, G2. What are the purposes of G1 and G2? Just to hang out? Yep.
Or cell growth.
Cell growth, yeah. What else? So you've got G1. So G1 is happening after M. So that would be like some cell growth, but what else? What else is happening here? Before you replicate your DNA, what do you want to make sure of? That it's not damaged, right? If you've got thymidine dimers, that'll be bad. You want to make sure that the proper environmental conditions are there, so you have all your nucleotides and all your things that you need for S phase. G2, yep, cell growth, because you're going to have to grow in size before you can divide into two, right? Remember we had an extra credit about that at one point, about the amount of plasma membrane and how it doubles. What about G0? Does anyone remember learning about G0 at some point? It's like a timeout. Yeah, no, maybe. Some cells go into G0 permanently. What cells go into G0 and they stay there forever? Huh? Nerve cells, neurons. They definitely cells that don't divide frequently, you would say, are stuck in G0. Eggs, right? Eggs are stuck in G0 until they become fertilized. then they leave. If they stay in G0, they don't become a baby. Okay. Okay. So cell cycle is tightly regulated, right? And it's controlled by a whole bunch of proteins that we're going to learn about that are acting as biochemical switches. So these proteins are acting as switches that allow the cell to progress to the next phase. But only if the conditions are right, only if the previous phase was properly completed. So you can think of cell cycles being regulated at 3 main checkpoints. So there's the late G1 start. Where is that? Right there. Right? So is the environment favorable to proceed to S phase? If you don't get past that checkpoint, you're going to arrest prior to S phase. Then there's your G2M transition, right? You don't want to enter mitosis if there's a problem that happened during S phase, like a stalled replication fork, for example. So is all the DNA replicated? Is the environment favorable? And then there's your metaphase anaphase transition. That's a huge one, right? You do not want to proceed. What's anaphase? What happens to your chromosomes then? They separate, right? You do not want to separate your chromosomes if things aren't right. Like one of the main things are the chromosomes attached to mitotic spindle. If your chromosomes are not properly attached to your mitotic spindle, do you want to proceed to anaphase? No, right? You're going to leave a chromosome behind, and you have an uneven number of chromosomes in your daughter cells, and that's a really nice thing that sometimes results in cancer. And well, you see it. It doesn't result in cancer, but you see it frequently in cancer. So that's a big one. Or just if your cells, or if your chromosomes are just attached incorrectly, right? If your mitotic spindle, remember you're coming from both sides, if microtubules from each side attach to one of the sister chromatids, right, and then pull in opposite directions, you're going to break your chromosome, right? So these are all things that are going to stop the cell cycle from progressing. These are the checkpoints. Okay, so I keep saying cell cycle is highly regulated and it's controlled by many different proteins. What do I mean? Why is cell cycle so important? Why is it so important to be regulated? And it's not just like an automatic thing that just happens like clockwork. Yes? Right, so if it's not regulated, the most obvious outcome is cancer, right? When things, and most of the, almost every single cancer I can think of where I know something about genes that are dysregulated, it's usually genes that are associated with cell growth in some way or regulation of cell growth. Either a growth factor receptor or kinases. Remember RAS. And then the other thing is another class of proteins that are often dysregulated in cancer are proteins that are involved in inducing apoptosis, right? So there's like, you can think of it as a balance, right? You have cell cycle and then you've got apoptosis. And you need them to be balanced because if cell cycle becomes dysregulated, apoptosis needs to be turned on to kind of prevent these cancer cells from becoming cancer cells and spreading cancer to the whole organism. And if apoptosis, the genes regulating apoptosis are dysregulated, then you're really in trouble. And that's usually what happens in cancer. You get dysregulation of cell cycle and dysregulation of apoptosis, so you can grow uncontrollably and not die. So the question is, how do cell biologists study cell cycle regulation? And we're going to talk a little bit about that. Yeah. So there's most of the research done on cell cycle was done in yeast or xenopus, mostly just model organisms that are easier to work with than mammalian cells. So the most common model organisms are yeast, xenopus, that's a frog, and then also we do a ton of work in cultured mammalian cells, so cell lines that grow indefinitely. And the reason we can use model organisms is because the machinery that's involved in cell cycle regulation is highly, highly conserved throughout eukaryotes. So anything you learn about cell cycle regulation in yeast, you can probably apply to human. Same with frog. And that's what this week's recitation paper is about, right? Does everyone read it? Everyone's ready? Hopefully all of you who I'll be seeing shortly have read it. It's all about a system where they used a deletion mutant in yeast to identify a cell cycle regulation protein in human through complementation. So for those who have not yet read the paper, this might help a little bit more. So there's two types of yeast that are commonly used model organisms for cell cycle research. And they're used just mostly because of ease of genetic manipulation, and also they're cheap and easy to grow. If you want to do mammalian cell culture, it's really expensive. Each time you pass your cells, you need media which contains FBS, which is like over between $100 and $200 for a 500 ml bottle. It gets really expensive. But yeast you can just grow on agar or in broth like bacteria. It's really cheap. And they're really simple to genetically manipulate and make knockouts. And you'll be learning a little bit more about that in recitation. So there's two types. There's fission yeast, which splits through fission. And there's budding yeast, which splits by budding. And the type we're talking about in this week's recitation paper is in S. Pombe. So a lot of the early studies on cell cycle were done through analysis of genetic mutants that are defective in cell cycle progression. And the way that they're studied is you have a way to control when you can see the defect. And most of these this is called conditional mutants, and the most common is a temperature sensitive mutant. So when you have your normal temperature, everything works fine. But when you raise the temperature to the restrictive temperature, whatever gene that's defective will become defective, and you'll be able to see your defective phenotype. So for example, you can have a mutant that at the normal temperature will go through G1, S, G2, M, so normal cell cycle. And then at the restrictive temperature, it stops right here, right? It stops before S phase. So whatever gene is mutated, you could say is probably involved in the start of S phase or the start of DNA replication. And if you wanted to study it, you would, like in the paper that we're doing this week, they use a mutant just like this, and they complement it with a whole bunch of human genes and wait. And they basically look at how the cells are growing, and once they see the cells that no longer are arrested, not at G1S, but I think it's somewhere M phase, then they know what the function of the gene that they put in is, because it complements the defect. Does that make sense to people? It will by Friday. Okay, so here they use budding yeast to study defects in CDC15. So they're the same kind of mutant that grows normally at a normal temperature, and when you increase the temperature so that you can see the phenotype of the mutant, then you see it grows like this. So let me turn the light off. Okay, so here's wild type. Here's an increased temperature, so you can see that the actual phenotype of the mutant is able to be seen. And this is how they would look at these. They would literally look at how the cells are growing and decide whether or not there was some kind of defect, and they'd isolate those and they would study those mutants. So what point of the cell cycle do you think is being affected here? cytokinesis. So there's probably something having to do with the metaphase to anaphase transition, or just cytokinesis. Yep. So this is a defect in CDC15. So what do you think CDC15 does? Okay, so it could have to do with it forming the contractile ring if we're similar in these cells. It could have to do with anything pretty much after metaphase all the way to cytokinesis, right? Because cytokinesis is not being is not being accomplished. That was nicer. Okay. Any questions so far? Everyone understands the concept of using a mutant to study something to study cell cycle, and how you can physically see this, right? You can see that it's not splitting. And in this week's paper, you're seeing a similar thing. Okay, so other model organisms are Xenopus, Spatula, and Drosophila, and they're useful mostly because they use the early embryos. They have very large cell size. It's easy to manipulate. An example from Xenopus, you can actually take the cytoplasm from the egg. These eggs are huge. And nuclei from frog sperm, add ATP, and you can actually watch cell-free mitosis and study mitosis that way. And then you have your immortalized cell lines. These are cell lines that grow indefinitely. They're usually transformed in some way, either by some kind of viral gene or some mutation in oncogene, and that's what allows them to grow indefinitely. Primary cells, which are not cell lines, those are the cells if I took cells off of your skin right now and tried to grow them in a dish, they wouldn't grow many more than, I don't know, 20 divisions or so. And that's because you don't want them to, right? If I took cells off your skin and grew them in a dish and they kept growing, what would that say about your skin? You're probably in trouble. You're probably going to have some sort of cancer, and you'd thank me for that early diagnosis. And mostly it's because unless you supply different growth factors, cells shouldn't grow. So these are tools for studying cell cycle in mammalian cells. You can directly visualize cells, right? Cells in mitosis are rounded. Cytokinesis is easily visualized. You can look right at them in the microscope and see how many times have I shown you immunofluorescence pictures where you've seen a cell in mitosis, in some phase of mitosis, right? You should be able to visualize it in your head right now. Who can do that? Everyone, raise your hand, right? Usually the DAPI is staining the DNA, and sometimes the nuclei are like this, right? Sometimes your chromosomes are condensed, right? That's very different, right? So you've got interphase cells. You've got two phases of mitosis here, right? This is what? What do you think that is? You think this is earlier than this one? Prophase, probably. Your chromosomes are starting to condense. And then here your mitotic spindle's already formed and your chromosomes are lined up in metaphase, right? So you can just look at them either by without antibodies. Here you're seeing the beginning of cytokinesis, or with antibodies or DNA stains, right? So this is DAPI staining the DNA, and these are antibodies for what? What antibodies did I use to see these long stringy things? You're all fired. I fired every single one of you just now. Can I fire my students? What antibodies would I use to visualize these long line things? Yes. GFP, no. So, okay, so maybe you guys are missing what an antibody is. So, an antibody is going to bind to a protein, so you can visualize it. So GFP would be a tag that you would attach to protein and another way to visualize things. But if I want, so what are these? Does anyone know? Huh? It's a what part of the cytoskeleton, people who just took a test on this? What happened? Did you guys eat too much pumpkin pie? Huh? Yes, microtubules. Great. So if you wanted to visualize your microtubule, you would go to Abcam or whatever company sells antibodies and buy an antibody against tubulin. Right. Tubulin, microtubules. You guys knew that. Come on, people. You guys are good. I know it. I saw those exams. Okay. Right? So your mitotic spindle is made of what? Microtubules, right? So these are, okay, I'm going to stop. Okay, let's move on. Cells in S phase can be identified by detecting incorporation of base analogs with specific antibodies. What's a base? Who wants to guess what a base analog is? Yes. Right, a different version of a nucleotide, exactly. So BRDU is a very common one that is used. Bromodeoxyuridine, and it's a thymidine analog. So everywhere T should be incorporated during DNA replication, BRDU is incorporated. And the purpose of that is so that then you can use an antibody against BRDU, and you can see it. So you can see cells that well, let me ask you guys. So you are studying cell cycle, and here's your DNA, very short DNA. Here's the DNA of your cell, and as you have replication, right? Remember, you have semi-conservative replications. So one strand of your DNA, all the T's are going to be fluorescent green, okay? Because it's incorporated BRDU at every spot where T should be. And then you've treated your cells with your antibody and put your secondary, now it's green. What does this mean about these cells? What have these cells recently done? What phase of the cell cycle have these cells recently gone through? If they have, so you take a picture of your cells, right? And you stain all your cells are red, but your green cells are the ones with BRDU. They're BRDU positive. So what phase of the cell cycle did those green cells just go through? S phase, right? Because when is DNA replicated? When else would you be able to incorporate a thymidine analog, right? Only during DNA replication. Because it's not going to just remove thymidines and replace it. It's incorporated by DNA polymerase. Okay? So this is a really great way to study cell cycle because now you can detect all the cells that are in S phase, right? Okay, so researchers study cell cycle by adding the thymidine analog BRDU to cells. BRDU is incorporated into newly synthesized DNA. BRDU positive cells are likely to have what? A, right? Recently progressed through S phase. That's the only way you're going to incorporate a base into your DNA is by replicating it, right? Okay. Okay, so let's talk about flow cytometry. Who knows what flow cytometry is? It's okay. You're not supposed to. Okay, so flow cytometry is a way to count individual cells. You can think of it that way. And you can count the individual cells based on anything you want. You can count it based on size. You can count it based on fluorescence. If you have cells that have GFP tubulin in them, you can see how many have GFP tubulin by counting green cells. So this is what flow cytometry, a flow cytometer kind of is made of. You've got this, something that's going to allow the cells to go through. So there's a nozzle, and then there's lasers, and then there's a whole bunch of mirrors and detectors that send a signal to a computer, right? Because it's all about a computer these days, right? And the point is that the cells drip cell by cell through, so it's a stream of liquid that's going in front of the laser, and the laser is going to hit the cells, and the light's going to scatter in a certain way. And based on how the light scatters when it hits the cells, right? If I shine a flashlight at like a really skinny little person and like a morbidly obese person, would you say that shadow would be different? Maybe. That's the easier way to understand it, right? So there's cells of different sizes. Some are little mini people, and some are morbidly obese cells, right? Anyways, so the light's going to scatter in different ways, and it's going to be detected in different ways based on whatever you do to it. So I've done this before in the past because I wanted to look at the effect of some protein on infection of cells with adenovirus. And when the cells are infected with adenovirus, they were green because it was a GFP virus. And so when I added my protein in, it altered how many GFP cells there would be. And so I put it in the flow cytometer, and it gave me a percentage of total cells. So I'll say, count 5 million cells. And it will say 10% of your 5 million cells were GFP positive, meaning they were infected with my virus. So I'm going through this because it's useful to know if you're ever going to do any kind of research. It's useful to know if you're ever going to do any kind of reading of journal articles, especially in immunology, because immunology, they love flow cytometry because they're all about counting cells that are expressing different surface antigens. And it's very useful in cell cycle analysis. So when you detect all your cells and all these mirrors and detectors and whatever send a signal to the computer, you'll end up getting, you'll basically either get a plot or a peak that tells you what your cells are doing. So in this case, it might be number of cells with different intensities or number of cells over time. This one might be different colors, GFP cells or red cells. And so we'll go through a little bit more of that. Okay, so why am I rambling on about flow cytometry? Well, being able to analyze cell cycle by looking at DNA content, well, that's a really good way to analyze cell cycle, and doing that requires the use of flow cytometry, right? So in S phase, prior to S phase, how much DNA do you have? Like 1 time as many or 2 times as much? Right? There's like one, right? So there's one set of chromosomes, right? And what happens? What happens at the end of S phase? Do you have the same amount of DNA? Twice as much? Twice as little? Twice as much, right? Because you've doubled, during S phase, you double your total DNA in your cell. Does that make sense? Right? Has anyone ever thought of S phase as a doubling of total DNA? Maybe? No? Yes? Someone did. That's good. Thank you for participating. I love when people participate. Right. So, and then you have M phase. So you have S phase, and then your G, what is it? Your G2, and then you go into M phase, and then you split, right? So right before cytokinesis, you have double DNA, and then when you split, how much DNA do you have? So you still have double DNA? You guys are looking at me like I'm like a 4-headed monster up here. Who wishes it was still Thanksgiving? Who wishes they were at the parade right now? No, it's cold at the parade. I went one year. It's cold. Hot chocolate is expensive. You just want the hot chocolate to warm your hands, really, because no matter what gloves you're wearing, your hands are cold. And then the hot chocolate's like $5 because you're in New York and they know that you want to warm your hands. Okay, so the point of what I'm saying, right? So I want you to look at this graph here. Okay, this is right basically where I took that question on the quiz from. So this is the amount of DNA per cell, right? In G1, you're going to have this much. In S phase, it's going to increase, right? Because this is increasing. Lower amount of DNA, higher amount of DNA, right? Increasing. S phase, it increases. And then G2 and M, it gets to its max until cytokinesis, right? So at any given moment, this many cells are in G1, this many cells are probably about in S phase, and this many cells are in M phase. Okay? So you can analyze this by flow cytometry by staining your DNA, right? Because if and you'll use many different stains, but the most common is propidium iodide, which makes the DNA orange. And if you have one set of your chromosomes, then you should have a certain amount of staining intensity. If you double that, your staining intensity should double, right? And you can measure that by flow cytometry because your detector, you have set your detector to detect orange intensity. How many of my cells have this much orange? How many of my cells have twice as much orange? Right? So cells with the highest amount of fluorescence, the most orange, have the most DNA, so they have double DNA, so they've gone through S phase and have not yet split into two. Okay? And so this is very commonly used to analyze cell cycle because you can see it all in one, right? Just by counting how much or quantifying how much DNA you have, you can analyze anything about cell cycle. Let's say you're studying a drug that's going to halt the cell cycle. Let's say you're studying a mutation that's going to activate a checkpoint. All of these things. Let's say you're studying a virus. I did a study once where we were looking at an HIV protein that was involved in cell cycle arrest. So what did we do? We did exactly this in the presence and absence of our viral protein, and we saw a difference. Okay, any questions about this? I feel like I'm talking too much today. Should I stop? Yes. No. Okay, fine. I'll keep going. Questions? Does everyone love PI staining? I think PI staining is pretty awesome. Orange. Who doesn't love color? Okay. So this is what it could look like, right? So you can either do a simple staining of, this is just staining cells, HeLa cells, with PI, which is going to stain your DNA, and antitubulin. And that's what it will look like. That is what is dropping through your flow cytometer, and each one is being counted at the different intensity. Now, mind you, you can't see much of anything here, right? Well, these cells are clearly not mitotic, right? Would you all agree they're not mitotic cells? Right. But that's what it looks like. And here, this is what it looks like with flow cytometry. So in this figure, Jurkat cells, so just a different kind of, it's a T cell, they were double labeled, so two different stains with anti-phosphohistone H3 and PI. Who's taking 409 right now? Anyone? So what is anti-phosphohistone H3? What is that going to detect? Other than that, it's physical. Okay. So phosphorylation of histone H3, it correlates with chromosome condensation. Remember, your histones and their modifications are going to say a lot about the chromatin structure and function. So when your histone H3 is phosphorylated, that usually happens during chromosome condensation during mitosis. Don't worry about that. You're not taking 409 with me right now. And then it's also PI. So you've got your DNA labeled with PI plotted here. And then you've got your histone H3 phosphorylation staining here. So you're looking for phosphorylated histones and DNA quantity, right? So closer to the 0 means less orange. Closer to this end means more orange. So the cells right here, what phase of the cell cycle, which each little dot is a cell, by the way. The cells right here, which phase of the cell cycle would you say they would be in? If they're at this end. So they have very little PI staining. What phase of the cell cycle do you not have lots of DNA? G1. Interphase. All of interphase? No, not all of interphase. No. Because S phase is where you double, right? Right, so basically, these cells over here in the middle should be S phase. So you have your 1x DNA all the way to your 2x DNA, right? As your intensity increases, you're going to move along this axis, right? And so then and it's useful that they put in the phosphohistone H3 because those you know are mitotic cells. So it's logical that they're going to be all over here, right? Now clearly they have not hit cytokinesis yet because then they shouldn't have twice as much DNA, right? This is at the high end of DNA. Are you guys understanding how I'm reading this? Please, someone stop me. Yes?
So based on what you're saying, then the blue cells underneath where the mitotic cells are in G2? Because they have more DNA, but they don't have histone.
Yes, that's a great way to think of it. Okay, yeah. So here's your mitotic cells because they're being stained for histone H3, and they have lots of DNA. But the ones that do not have their phosphohistone, they're going to be just prior to establishment to beginning of mitosis. Yeah, and so somewhere in the middle here, that's your S phase, right? Because you have your increase in your DNA content, and over here you're going to have what? What's before S phase? G1, right? Or yeah, G1. Okay. I don't believe anyone here feels good about this. Someone convince me. It's different to look at, but once you get it, it's really easy. So you just got to get it. So everyone think about it, look at this, talk to your friends, and then ask me questions if you don't get it. Okay. And so that goes to this question, which if you read it and were reading the book, you would have come across that figure and been like, that's what she's talking about. So in order to study cell cycle regulation, researchers need tools to evaluate what phase of the cell cycle a cell is in. One tool is to stain the DNA in cells with PI and use flow cytometry to count the number of cells with different PI staining intensity. PI staining intensity will vary at different phases of the cell cycle due to the differences in the DNA content per cell, right? So you're working with a drug that should arrest the cell prior to completion of M phase, right? So how much, if that drug is working, how much DNA should be in a cell if it's arrested prior to completion of M phase? One set of chromosomes or two? Two, right? Because your cell hasn't divided yet, but you've duplicated your DNA. So you're going to have twice as, you're going to be stuck with twice as much. You expect the following results compared to control untreated cells, right? Because you always have to compare to untreated. Right, so control cells will have 60%. So the way to approach this question would have been to go like this. Okay, you got 60% here. Here's a graph. So you have 60% of staining at 100%, and 10% somewhere over here. And 30% at 200. So that's your control. And so you know that your PI staining doubles, right? So you know that this is your S phase, and you know that this is pretty much G2M, right? And this is G1. So that's your control. Treated cells will have 100% at 200. So if you were to put your treated cells in, what you want to do before you even look at these confusing answers is think to yourself, well, if I'm arresting cell cycle prior to completion of M phase, so I should have twice as much DNA. My staining intensity should be about up here, right? Because it's all going to get stuck right here. Right, so that's control. drug. And that's the answer you should have been looking for, because it's being stalled there because you've treated with a drug that won't let it get past there. So that's how you approach that question. I noticed people had a little bit of trouble with it. Does anyone have questions about this one? Now on an exam, I would show you a picture. Definitely make sure you understand these kind of questions. Okay, 5 more minutes. Is that true already? We've been having so much fun. I'm so glad you guys are here today. Okay, so let's talk about regulation of cell cycle control. So we just talked about all the different ways that scientists study cell cycle, and it really has to do with counting cells that have certain amounts of DNA, basically, or looking at incorporation of BRDU. Those are really obvious ways. And then also looking at mutants in yeast. That's another nice one. So how is cell cycle regulated? So pretty much it all has to do with CDKs, cyclin-dependent kinases. What does CDKs do? What is their main basic function? Phosphorylate, yes, because they're kinases. So if you had to guess, what do they phosphorylate? General, big picture. Don't give me a protein name. Things that need to be regulated during cell cycle. Okay. They phosphorylate different proteins that need to be turned on and turned off at different phases of the cell cycle. Okay, that's how cell cycle works. So kinases control the activity of many different proteins involved in controlling the major events of the cell cycle. And we're going to go through a bunch of them. So you've got your CDK, and it's regulated by this cyclin. So when the cyclin binds to the CDK is active. When there's no cyclin, the CDK is off. And if you think about it, you've got G1, you've got S, G2, you've got M, right? You've got a lot of phases of the cell cycle that need to turn on and turn off at different times. It all has to do with the activity of these CDKs, which has to do with the presence of this cyclin. Right, so basically what happens is cyclins rise and fall throughout the cell cycle and that activates the appropriate CDKs that need to be turned on at that appropriate phase. So it's all a pattern of increased transcription, translation, and then degradation via ubiquitin proteasome when it's no longer needed, right? So you'll have G1S cyclin come up here, and then it'll be degraded at the beginning of S phase, right? So that will turn off that CDK. Then you'll have S phase, increased transcription translation, high levels of S cyclin, so the S CDK will be on, and then it will be degraded and turned off here, right? You need that because you need to transition out of S phase and into M phase. Your M cyclin, it'll go up before M phase because you need that buildup of M cyclin to activate your M CDK so you can enter M phase. And so whether or not those cyclins are there are going to determine whether that kinase is active and whether the appropriate proteins can be turned on so that you can progress into the next phase of the cell cycle. Okay? Very, very basic concept that you will die right now if you don't get. You will die a horrible, slow, torturous death on the final if you don't understand this concept. Right. Okay. I guess we have 1 more minute. Should we do 1 more slide, or do you want to leave? Okay, 1 more minute. Okay. So there are many different... Sorry. It's the end of the semester. I'm feeling a little evil. Okay, go. Leave.